Hello, this is Muhammad Umar Farooq Biological Video. Please make sure to subscribe, like and share. Ask the question for answer and comment about the artwork. You can also answer question to each other for solving the problem. So today we will discuss about the previously we was talking about the anatomy and the structure of the coniferals, while here we will discuss about the reproduction. So here is the mistake about that. It is the male and female cone are present in the same plant, but that was my mistake about that, so you can understand as well. So the reproduction of Pinus is the Heterosporus. Heterosporus means that two type of spore will be produced from the Megasporangium and Microsporangium. So it means it is contain the Megasporangium and Microsporangium. So the mega mean bigger than the Microsporangium and micro means smaller. So let's talk about here is the reproduction we will discuss. So the reproduction of the coniferals, that is the Pinus. So the develop micro and megasporophiles, which that is developed in the same plant, and same plant develop both cones, male and female. So here is we will discuss about first of all the male cone. So the male cone is looking like this, and here is the diagram of the cross section of the male cone in looking like this. And that's when we will separate this one microsporangium. So this microsporangium contain a microspore. This microspore is contain different layers. So it means the many spores are present, which that is inside is the microspore mother cell. So this microspore mother cell will be divided. So and hypodermal layer are present, and the inside of the hypodermal layer is the tapetum. So the tapetum is important for the providing of the nutrition, and cell layer reticulate help in dehiscence and the upper layer. So remember about that. So while it's after the division to form a tetrad, so I am drawing the three celled after divided, but it is cross section, one is not show in this diagram. So the four layer sporangium wall will be formed. So this wall will be formed, and archisporangia cell, sporangial cell is wrapping around these spores. So after this development is look like this. This is the early wings or air bladder or wings, you can say, which will be developed. It is the four, and it means that here is the adult form of the spores, which contain inside is the nucleus, which that is in the form of sperm, you can say. So the axine and intine is the layer, and wing are present for using for the flying into the air to trap into the female cone. So that is the microspore, and here is the nucleus, which that inside is the microspore. So this microspore is important character, while here we will discuss about the female cone. So the female cone is basically is look like this. This is the bigger, and this contain here is the needle like structure of leaf, which that is attached with the spurs, and that is known as the Megasporangia because it contain a megaspore. So remember about that. And it means here is the bracket, which that is showing in the figure, and here is I am drawing the cross section of the Megasporangium, which contain a ovuliferous scale, and this ovuliferous scale is inside is the egg. So their development of this egg is downwards looking like that, which that is a new one will grow in the upside, while the downward it grow, it is the development. So here is the ovule, and this is known as ovule, while here is the pollen chamber are present for trapping the basically microspore, and that is the integument is here. So this is the true bracket like structure is the cross section, and that is the pore is known as the micropyle, and I am drawing the whole diagram to upper part is looking like this. And this is the here is basically here is the tube nucleus are present trapping inside, while that is the pollen tube, and micropyle is are present in the form of two brackets, which that integument is contain a micropyle looking like that, and downside is the bracket, you know. And here is the basically that is a pollen tube, which that is used for the multiple microspore, not single. So remember about that. It is the pinus which contain ovary which contain more than one archegonium, while on the other hand many plant will not do like this character. So its wing containing microscope will move inside from here. So there are many developing pollen tube. So the archegonium is here, and the prothallus female are present, which that is contain archegonium. So let's talk about the zygote after the fertilization of one archegonium with attached with the egg is containing, and zygote will be fertilized. Next video we will discuss about the whole detail of the reproduction of pinus. So this is the overview. So here is the development archiconia. So this development of archiconia is looking like this. It means the archigonial initial will be formed, and this archigonial initial is the A, looking like this. And after the development is the it's grow, and the downside is the cell is growing more. It means that it is the precursor. This cell is the produced by the cell division, and that is the diploid, and but the egg is the haploid. So it means it is the development to create a jacket. So that jacket is wrapping around the egg, or you can say that is the jacket is wrapping around the archegonium. So that means it is the protective layer. So remember about that. And upper part is the closing part, which when rupture during the maturation of the egg or maturation of the archegonium. So archegonium is allowed to trap the microsporangia for fertilization, while the microsporangia is the motile in the air, and archegonium egg is not motile in the air. So that means archegonia is egg is waiting for the fertilization. So remember to that. It is easy to understand. So the zygote is after that cell division to form a bicell and tetrate, and they basically 8 cell, and that is growing downside, but the cell division is moving upward. So that's create the force, and the embryo cell are present and secondary, and there are many embryos due to the more than one fertilization. So that is the rosette cell are present with that upper part, while here the suspensors are present. This suspensor is allowed to move inside, and it means the move forward due to the cell division backward. So that's produced like structure, this structure secondary suspensor. So this is the secondary suspensor. So this secondary suspensor will allow to move more, and that is in the form of cap, and there are a lot of basically embryo formation, basically a lot of cotyledons, not two cotyledons like other seeds. So this is the a lot of cotyledons so produce, and after this development to one cotyledon is single but into the plant. So thanks for watching. Please subscribe the channel.